While well-run research studies are powerfully positive tools to learn about the natural world, it is important to always recognize the restrictions that experimental design limitations, statistical errors, bias, and confounding make on the conclusion one wants to draw. I believe that within human nature we want to extrapolate beyond the limits of our data and to reach firm conclusions even when the available data is incomplete and imperfect. This aspect of human nature must be battled in order to have an accurate understanding of the natural world and to allocate resources most appropriately to improve animal and human health. Even though vigilant application of the scientific method and appropriately limited conclusions drawn from individual trials and systematic reviews is the process that should be used by epidemiologists to aid society, the medical and veterinary literature is filled with examples of poor study reporting, even for the gold standard of research to test the efficacy of interventions, the randomized controlled trial. Often these studies were designed or conducted poorly, but sometimes the study was conducted properly, but the report was so incomplete as to leave the research consumer unable to know whether to accept the findings or to throw the paper in the trash and erase all memories of reading it. A study done by Chan and Banduri in the Journal of Bone and Joint Surgery, published in 2007, assessed all the non-pharmaceutical randomized controlled trials from 8 influential medical journals over a 2-year period of time for important quality indications such as appropriate allocation concealment and blinding of patients, rehabilitation staff, and clinical outcome assessors. They found that 84% of the studies did not report how patients were allocated to treatment adequately for the reader to determine if the method was appropriate or not. Follow-up interviews with the authors of the studies revealed that 41% of those unclear studies actually had adequate allocation schemes. But that leaves 50% of all the trials published in the influential peer-reviewed journals in the study having inappropriate allocation of study subjects to treatment. They also found that 46% of the studies did not report whether or not study participants were blinded as to the treatment group for each patient. Further investigation revealed that 28 to 40% of the studies did blind the patient, staff, or outcome assessor, but that results in the disturbing conclusion that about 32% of all trials in this study did not blind important study participants to the treatment group of each study subject. This and many other studies that found similar results prompted human health researchers to find methods to increase the quality of study reporting in influential peer-reviewed journals. These efforts led to the creation of the Consolidated Standards of Reporting Trials, or CONSORT guidelines, for reporting randomized controlled trials. Many researchers and practitioners have recognized that it is difficult as a research consumer to assess the quality of published studies, even when published in influential peer-reviewed journals. The CONSORT statement allows anyone to grade the extent to which a report adequately reports important features of the study. The conclusions reached by studies that report strict adherence to appropriate randomized controlled trial design and conduct can be trusted to a much greater degree than studies that fail to report important study features, such as how subjects were allocated to treatment, how patients, support staff, and outcome assessors were blinded, how many and which patients were lost to follow-up, and how potential confounders were controlled. The CONSORT statement emphasizes the belief that authors must be completely transparent when describing the design and conduct of a randomized controlled trial, so that the research consumer can make informed decisions about the validity and acceptability of the research reported. Research consumers use the CONSORT statement by comparing a published article to a checklist for important study quality descriptors and a flow diagram of how study participants were selected, allocated, followed, and analyzed. The items for the checklist were selected because there is evidence in the literature indicating the potential for biased estimates of treatment effects when the items were not reported, or because the information was deemed essential to evaluate the reliability or relevance of the findings. Even though the vast majority of researchers intend for their experiments to be appropriately designed, conducted, and reported, even well-trained scientists with years of training and experience can make intentional, subconscious, or ignorant mistakes. As fallible humans, scientists can fail to recognize important sources of bias and confounding in the studies they design and interpret. They can be unaware of their level of ignorance on certain components of experimental design, statistics, and epidemiology that impact their studies. And they can extrapolate beyond the limits of the conclusions that can be drawn from a particular experimental design or study population. Therefore, the CONSORT statement, which provides clear guidance for complete and accurate study reporting, is needed to improve the conduct and reporting of randomized controlled trials. The need for the CONSORT statement is illustrated by the fact that many reports of randomized controlled trials either do not report how subjects were allocated to treatment, or they report an allocation scheme that is not random. In addition, many studies do not report that key personnel in the conduct and interpretation of the study were appropriately blinded. This reporting flaw can be obvious if the authors clearly indicate that treatment administrators, animal caregivers, outcome assessors, or any other person involved in the gathering or interpreting of study data are not blinded. Research consumers will also question the rigor of blinding used in the study if specific methods of blinding are not described. And finally, because some loss of subjects between the onset and completion of a study can be expected, it is essential that all RCT reports indicate how many subjects were allocated to each treatment but not evaluated in the final statistics. Anyone reading a report with excessive subject loss, even if it were evenly distributed between treatments, and anyone reading a study where even relatively minor losses were unevenly distributed between treatments, would be very concerned about the validity of drawing any clinical conclusions. For randomized controlled trials, the proper randomized allocation of study subjects to treatment group is a critical feature in the design and conduct of the experiment. And because of the critical nature of this study characteristic, the report must clearly inform the reader of the exact method of subject randomization. In a review of schizophrenia studies, it was found that only 4% of 2,000 studies that claimed to be randomized controlled trials clearly described the method of allocation. And the fact that a study claiming to be a randomized controlled trial is published in a highly respected, peer-reviewed journal, and the investigators are usually highly trained in their area of expertise, does not ensure that randomization and other study features are done correctly. In this study of effectiveness for prophylactic use of providone iodine in newborns to control neonatal conjunctivitis, published in the New England Journal of Medicine, the authors did not randomly allocate treatments to study subjects, but rather treated all newborns for 1 week, then used a different treatment for all subjects the next week. This type of allocation is definitely not random and may have any number of potential confounding factors that are not controlled. And as another example of non-random allocation in a study claiming to be a randomized controlled trial, a study published in Obstetrics and Gynecology allocated all subjects available in a given week to one of two treatments, rather than a random allocation scheme. Dickinson and colleagues evaluated 208 trials investigating head injuries published in reputable journals and found that 22 trials, about 10% of the studies, used adequate methods of random allocation, including telephone number, identical containers with treatment codes enclosed, random allocation schemes controlled by the hospital's central pharmacy, and sealed envelopes with treatment codes enclosed. They also found that 25 of the trials, about 12% of the studies, reported allocation schemes that were definitely not random and subject to selection bias. These included 18 studies with various improper methods, 1 study that used date of birth, 3 studies used day of the week, and 3 studies alternated treatments between consecutive subjects. The largest number of studies, 161, did not provide any information about how subjects were allocated to treatment. Another study characteristic that is essential for unbiased assessment of treatment effect is blinding. A study reported in the Journal of the American Medical Association found that studies that did not clearly indicate the method of blinding or that did not adequately use blinding exaggerated the effectiveness of interventions by an average of 30 to 40%. Another study investigating the importance of blinding found an even greater impact of about 70% exaggerated treatment effectiveness in orthopedic studies using continuous outcome variables where the outcome assessor was not blinded versus studies where the outcome assessor was effectively blinded. When studies using a subjective binary outcome, such as treatment success versus failure, assessment bias is even a greater concern than when the outcome is objective. A study published in the British Medical Journal in 2012 reported that the most common misclassification in studies with subjective binary outcomes was optimism error. In other words, unblinded assessors identified fewer treatment failures than blinded assessors. This bias was present for both treatment and control groups, but was more pronounced in the treatment groups. It is also interesting to notice that blinded and unblinded assessors agreed on a vast majority of patients in the various studies examined, and that the median percentage of differential assessments was only 2.5% to 3% of the study subjects, with a range of 0 to 28% differential between blinded and unblinded assessors. However, this occasional difference in treatment success classification between blinded and unblinded assessors resulted in large and clinically significant differences in assessments of treatment effectiveness. The study authors pointed out that although very few of the studies they reviewed adequately implemented blinding, there was nothing about the study design that prevented effective blinding. Because appropriate allocation of study subjects to treatment and blinding of outcome assessors is critical to control bias, a study in Lancet evaluated all the randomized controlled trials reported in 4 major medical journals and found that slightly fewer than 1/2 reported appropriate subject allocation. Only 26% reported appropriate blinding controls. Only 15% reported both appropriate allocation and blinding. I hope the last several slides demonstrate the critical need for improved study design and reporting. This identified need provided the drive to produce the first consolidated standards of reporting trials, or CONSORT statement. This effort started with the Standards of Reporting Trials meeting in Ottawa, Canada in 1993, and reached an important milestone when the first CONSORT statement was published in 1996. In 2001, a revised statement was published in 4 leading medical journals simultaneously: the Annals of Internal Medicine, the Lancet, the Journal of the American Medical Association, and the Biomed Central Medical Research Methodology Journal. This slide depicts the revised 2001 CONSORT as it appeared in the Journal of the American Medical Association. With continued input, the CONSORT statement was revised again and updated so that a new version was published in 2010. The latest version of the CONSORT statement, as it appeared in the Journal of Clinical Epidemiology, is depicted in this slide. The CONSORT statement has had important impact on the reporting and conduct of randomized controlled trials. Many major medical journals endorse CONSORT and require that all articles submitted for publication use the CONSORT checklist. Some of the topic areas for the checklist include title and abstract, introduction, and methods. The checklist continues with sections covering results, discussion, and other information. A person evaluating a published randomized controlled trial report can use the checklist to quickly and thoroughly evaluate how well the authors reported each of these important components of a well-designed, appropriately conducted, and clearly written study. As you might expect, as researchers became more familiar with what is expected in a study write-up in journals that have endorsed the CONSORT statement, more attention to appropriate study design and conduct is an added benefit. Because selection and allocation bias are extremely important to avoid for a study to be considered high quality, the CONSORT statement recommends that a diagram depicting the number of study subjects randomly assigned to each treatment, the number that actually received the intended treatment, and the number of subjects included in the final analysis be clearly communicated to the reader by the use of a flow diagram. The explanation for the diagram should include a clear depiction of the number of exclusions and losses from each treatment group and the reasons the subjects were excluded or lost. This slide depicts a participant flow diagram that is extremely helpful to readers to fully understand the efforts made to avoid selection and allocation bias, as well as potential breakdowns in the actual conduct of the study. Subject enrollment should be addressed in every randomized controlled trial by indicating the number of subjects who were available for the study and how that number was reduced to the final number of subjects in the study. The authors should report how many potential subjects did not meet the inclusion criteria and how many refused or were otherwise unable to participate. Next, the authors should indicate the exact number of subjects who received each treatment and a reason for any subject who was randomly assigned to a treatment but did not receive it. The authors should also report how many subjects were lost to follow-up or discontinued the treatments and the reasons for the losses. And finally, the flow diagram should clearly indicate the number of subjects that were included in the analysis. By comparing the number of subjects included in the analysis with the number enrolled and randomly allocated to treatment, one can detect excessive losses of study subjects or differential loss between the treatments, that is, more losses with one treatment than the other. Although there have been more published studies raising concerns about the quality of randomized controlled trials using human subjects than animal subjects, I am not aware of any reason to think that researchers investigating animal health do not make the same mistakes as their human health counterparts. In fact, Jan Sargent and others reported important problems with the methodologic quality and completeness of reporting of livestock field trials. They found deficiencies in how and if important trial features were reported, with only about 2/3 of the randomized controlled trials reviewed for their study reporting key features of the randomization strategy. Only 4% reported that double blinding was used, 62% reported the number of subjects lost to follow-up, and 91% of the studies had more than one study outcome, with the range being 1 to 41 outcomes for the studies evaluated. What is very important is that almost none of the published studies that had multiple outcomes reported which was the primary outcome of interest and which of the up to 40 other outcomes were not considered in the original design of the experiment. We all recognize that one can only use one or at most a couple of outcomes when considering the number of subjects needed, the type and timing of sampling, the number of study subjects housed together, and other allocation, sampling, and interpretation limitations. Because studies are not optimally designed for secondary outcomes, no or very limited conclusions can be drawn from an analysis of secondary outcome data. These types of analyses should only be used for hypothesis generation, not conclusions. It is also concerning that less than 50% of studies with multiple observations taken from the same study subjects over time accounted for the lack of independence of the measurements. Similar to studies of the human health literature, Sargent's study also reported that the extent of reporting deficiencies of a published paper were associated with the likelihood and magnitude of positive treatment effects. In other words, studies that carefully reported trial design attributes such as allocation schemes, blinding strategies, and statistical analyses were less likely to report a positive treatment effect than published articles that had incomplete reporting of key study design characteristics. For example, a systematic review of trials evaluating the efficacy of vaccination for the prevention of pink eye in cattle by Burns and O'Connor found that trials not reporting blinding and random allocation to treatment group were more likely to conclude that the vaccine was efficacious than trials where the study features were reported. In response to the recognition that randomized controlled trials of animal health topics suffered from similar reporting and methodologic weaknesses as human studies, a group of veterinary researchers and journal editors constructed a CONSORT-like statement that followed the groundwork of the CONSORT statement, but also addresses RCT features that are unique to animal studies. The reporting guidelines for randomized controlled trials in livestock and food safety, the Reflect LFS statement, was published in 2010 with the goal to improve the reporting of randomized controlled trials from veterinary medicine and food safety research. Although the CONSORT statement provides valuable guidance for animal health researchers, the authors of the REFLEX statement understood that there are some important differences in randomized controlled trials conducted on human subjects and those conducted on animals or food products. Compared to trials using human subjects, animal studies have two types of participants: the owner or manager of the animals who first agreed to participate in the study, and second, the animals that are the actual study subjects. With animal and food safety research, because of the common use of group housing and group allocation to interventions, the organization structure are often hierarchical and complex. These complex organizations lead to non-independence or clustering of observations. A specific example is when one has animals nested within pens, pens nested within rooms, and rooms nested within herds. In addition, repeated measures over time are often collected on the same study subjects, creating another opportunity for non-independence of study outcomes. Deliberate agent challenge is a common design feature of animal randomized controlled trials, but it is not acceptable in human research. And finally, the outcomes of animal studies are often non-clinical. For example, the outcome may be contamination with a foodborne pathogen that does not cause clinical disease in the animal reservoir, or the primary hypothesis may be that an intervention results in a different production of milk or meat as measured by daily pounds of milk production or weight gain compared to controls. The REFLEX statement collaborators started with the framework of the 22-item checklist for the CONSORT statement that had been published in 2001. 14 of the items were modified for the REFLEX statement, and one sub-item was added to address challenge study designs. In addition, some wording changes were added to address terminology differences between human-centric and animal-centric studies, as well as to communicate the hierarchical or clustered nature of common domestic animal housing systems, necessitating a description of the multi-leveled allocation schemes. The REFLEX statement was simultaneously published in 5 veterinary journals: Preventive Veterinary Medicine, the Journal of Food Protection, the Journal of Swine Health and Production, the Journal of Veterinary Internal Medicine, and Zoonosis and Public Health. The following slides report the 22-item checklist of the REFLEX statement. All the text in bold are modifications from the 2001 CONSORT statement. The first item in the REFLEX checklist is a recognition that the title should clearly indicate that the study was a randomized clinical trial, with a brief but clear description of how the study units were allocated to treatment. In addition, the title or abstract should clearly indicate whether the outcome was the result of natural exposure or from deliberate agent challenge. The term study unit was preferred instead of animal unit because it is common that a part of an animal, such as a hoof, teat, or eye, to be allocated to treatment, as well as individual animals, pens of animals, or barns or whole farms. The introduction should provide the scientific background and explanation of the rationale for the study. This should include a description of the nature, scope, and extent or magnitude of the problem under study, the pathophysiologic basis for the proposed treatment, or the justification for considering a new treatment regimen when there is an existing treatment, as well as any other factors known to influence the outcome and interpretation of data for the study subject. Item 3 reflects a difference from human-centric studies in that the eligibility criteria for both the animal owners or managers and the animals themselves should be clearly communicated. This is important to communicate because eligibility criteria may lead to meaningful differences between the study population and a population that a reader may want to apply the study results. In livestock trials, the concept of participant refers to the owner or manager of the animals who consents to participate in the trial. Thus, it is important to report eligibility criteria of the owner-manager and study units. Frequently, the only determinant of eligibility for a facility may be a personal relationship with the researcher. If such convenience sampling is used, it should be stated. At the study level, eligibility criteria commonly include age or production stage, sex, comorbidities, or previous treatments. The setting and location may affect the external validity of the study. It may be relevant to report the geographic location where the trial was conducted and the time of year when the study was conducted. Issues related to setting that could influence the external validity of the study should be described. Authors should describe the group sizes for all relevant levels of the organization structure, such as the total animal population of the facility, and the number, size, and capacity of barns, pens, or cages used to house the study units. Also describe feed and other pertinent management details and the presence or absence of the disease of interest or other endemic diseases. The unit of allocation for the intervention should be clearly stated, and this unit must correspond with the unit of randomization. The allocation unit can only occur at one level of the organizational structure. For example, in a swine study evaluating the impact of water-based vaccine on weight gain, barns may be randomly allocated to receive the water-based vaccine or a placebo. Therefore, the allocation unit is the barn. In a challenge study evaluating the impact of a chilling process intervention on the prevalence of Campylobacter on poultry carcasses, carcass halves may be randomly allocated to receive either processing method A or B. Therefore, the allocation unit is the carcass half. Pharmaceutical, biologic, surgical, food processing, or other interventions should be described with sufficient detail to allow readers unfamiliar with the products or procedures to replicate the intervention. If the intervention is applied to individual animals, the author should state whether the animals were individually housed or housed in a group. If housed in a group, the number per housing group should be reported. If the intervention was applied at the group level, the authors should clearly state the number of animals per group. The description of housing of the allocation units should correspond with the levels of the organization described in the participants checklist item number 3. In some challenge studies, non-challenged animals are included to serve as negative controls. When this is a feature of the trial, the number of negative controls and their housing relative to the study units, that is, within the same pen, stall, or cage as the challenged groups, or proximity to the challenged groups, and the opportunity for contact with challenged animals should be described. The precise details of the challenge model used in the study are critically important for assessing the external validity. The onus is on the authors to provide sufficient details of the model used in the challenge trial to enable the reader to assess its validity as a model for the real condition. The authors should clearly state the objectives introduced in item 2, the scientific background and explanations of rationale, and the corresponding null hypothesis to be tested. Sometimes the null hypothesis is that two treatments are the same with respect to the primary outcome. That is, when the study is designed to test the superiority of an intervention. While at other times, the null hypothesis is that the treatments are different with respect to the primary outcome, when the study is designed to test the equivalence or non-inferiority of an intervention. It is important to clearly state the null hypothesis so that the reader can correctly interpret the meaning of non-significance. If there are multiple objectives, the authors should characterize them as primary versus secondary, and if there are more than one secondary objectives, they should be ranked relative to their importance to the study's focus. The outcomes for a trial need to be linked to the objectives and hypotheses. All outcomes should be identified and defined, and the methods to measure each outcome should be described. If disease status is used as an outcome, a case definition should be provided, and the training of the person responsible for assigning that diagnosis should be described. If specific diagnostic tests contribute to the assessment of the outcome, include the sensitivity and specificity estimates, including a justification of why these sensitivity and specificity estimates are applicable to the study population. The primary outcome refers to the measure used to determine the study sample size. Other outcome measures, which may be equally important but were not used to determine the sample size, should be referred to as secondary outcomes. The rationale for differentiating the outcomes as primary and secondary is to allow the reader to understand which of the outcomes have sufficient power to detect meaningful differences, and therefore how to appropriately interpret the meaning of non-significance. In livestock trials, it is common to have one outcome related to the disease of interest, often reported as morbidity risk or mortality risk, and one related to performance, such as average daily weight gain or average daily milk production. In these circumstances, both outcomes are of interest, but because a binary outcome such as morbidity risk requires a larger sample size to detect meaningful differences than a continuous outcome such as weight gain, the disease risk outcome is used to determine sample size. Authors should provide details of any particular steps used to increase the precision or validity of an outcome measure. For instance, multiple samples or repeated measures may be used to define the outcome status. Limits of detection, precision of measurements, and cutoff points should also be described. Use of an adequate sample size to detect treatment differences that are economically and biologically important is fundamental to sound trial design. For the null hypothesis and primary outcome identified in items 5 and 6, authors should describe how the sample size was determined for each level of the organizational structure of the study setting. The descriptions should include how non-independence or clustering of the outcome measurements was accounted for in the calculations. The term random has a precise meaning, wherein each study unit has a known probability of receiving a given treatment prior to assignment of the treatments, and authors should specify the method of sequence generation, such as a random number table or a computerized random number generator. Deterministic allocation methods, such as alternate animal identification numbers, days of the week, date of birth, and gate cutting, are not random. When these methods are used, they should not be described using the term random or any variation of it. When authors do not use random methods to allocate study units to treatment groups, the method of allocation should be described in a manner that would allow the reader to determine if bias was likely to be introduced. Restricting methods, such as blocking or stratification, may be reasonable or preferred in challenge studies, as these types of studies generally use few study units. When sample size is small, random allocation may not result in treatment groups that are equivalent with respect to important outcome determinants. Nevertheless, random allocation within block or within strata should be used and described. The aim of allocation concealment is to prevent selection bias at the recruitment or enrollment phase of the trial. Allocation concealment differs from blinding, which aims to prevent misinformation bias in the measurement of the outcome and is implemented after allocation to the intervention. In order for the reader to evaluate allocation concealment, it is necessary to know who generated the allocation sequence, who enrolled study subjects into the trial, and how study units were allocated to the treatment groups. In controlled trials, blinding, also known as masking, refers to the process of keeping individuals involved in the trial unaware of the group allocation for the purpose of avoiding misinformation or observer bias. There are published reviews in both the human medical and veterinary literature that indicate that studies that do not report the use of blinding are more likely to report a treatment effect or a larger magnitude of effect than studies where the method of blinding is clearly described. It is insufficient to state staff were blinded to intervention group. The process of achieving blinding should be reported. In addition, 3 levels of blinding should be reported: individuals associated with the outcome assessment, individuals caring for the animals, and individuals involved in the data analysis. In challenge studies and some surgical studies, it may be difficult to maintain blinding. If the study cannot be blinded, the authors should describe why it was not and what controls were instituted to eliminate selection and misinformation bias. In addition, at least one objective measure should be used in the assessment of the intervention. A study that cannot be blinded and that only has an outcome, such as meeting a subjective disease case definition, has no control for misinformation bias and no validity. A complete and accurate description of statistical analyses allows the reader to assess the validity of the statistical methods and the likelihood of analytical bias. Particular care is needed when analyzing randomized controlled trials when the units of allocation and the units of outcome measurements are not the same. The assumption of independence and identical variance is commonly violated in animal studies when there are multiple repeated observations per study unit over time, or when study units, such as individual pigs, are aggregated into groups and the outcomes of multiple groups are considered in the analysis. Treating each observation as an independent event when the organizational structure of the population implies non-independence is a serious violation of inherent assumptions of many statistical tests and usually leads to an incorrectly low p-value. A randomized controlled trial, as compared to observational studies, is able to limit the potential for confounding by the use of randomized allocation of study units to treatments. Therefore, using multivariate analysis to explore confounding is not recommended, and if pursued, the rationale should be provided. If potential biologic interactions identified during the study design phase are of interest, it may be necessary to conduct subgroup or strata-specific analyses. If subgroup analyses are used, the method should be clearly described. Subgroup analysis of potential interactions identified post hoc is strongly discouraged. Authors are strongly encouraged to include a flow chart that includes all of the organization levels of the trial, especially when the complexity of the organizational structure is important to understanding the external validity when loss to follow-up or protocol failure occurred. Animal studies, especially those that are conducted outdoors under field conditions, may be influenced by seasonal and related weather effects. Therefore, the dates of the study and any unusual weather conditions that may be unique to one group should be noted. It is important for readers to know the characteristics of the study unit in order to evaluate external validity of the trial results. Descriptors for each treatment group, such as age, sex, weight, and parity, should be reported to determine the appropriateness of extrapolation from the study subjects to the target population. It is also critical that the authors clearly indicate the number of study units in each group included in the analyses, which serves as the denominator, and whether the analysis was by intention to treat, which is preferred, or an analysis of the differences between the study units that actually received each treatment for the length of the study. For each primary and secondary outcome reported in Item 6, a summary outcome should be reported for each intervention group. The reason to provide this information is to allow the reader to assess the clinical relevance as well as the statistical significance of the differences between the intervention groups. As the number of analyses using the same data increases, so does the risk of false positive findings. Examples of multiple analyses include evaluating the intervention against multiple outcomes, performing multiple analyses based on control of different potential confounding variables or within levels of an interaction variable, and subgroup analysis when interventions are evaluated within a subgroup of study units based on an important characteristic such as age or initial weight. Subgroup analysis can be planned during the experimental design phase of the study, which is preferred, or may be included as a post hoc decision based on preliminary analysis. If a subgroup analysis is only initiated after the study has been completed and preliminary analysis raises the potential for interaction, the post hoc nature of the decision should be clearly stated and the results of the subgroup analysis should be described as exploratory. When subgroup analyses are performed, information should be provided on the specific subgroups that were analyzed and the reasons for the analysis. All subgroup analyses that were performed should be reported regardless of the results. Bias may result from selective reporting of subgroup analyses. Many interventions have unintended or often undesirable effects in addition to the intended effects. Authors should clearly state all effects, whether or not they were intended or desirable. Interpretation of study results should follow the 5 recommendations adopted by both the CONSORT statement and the reflex statement, which were first published in the Annals of Internal Medicine by Moore et al. In 2001. They are 1. Brief synopsis of key findings. 2. Consideration of possible mechanisms and explanations. 3. Comparison with relevant findings from other published studies. 4. Limitations of the present study and methods used to minimize and compensate for those limitations. And 5. A brief section that summarizes the clinical and research implications of the work. Authors should address the biological and practical importance of the work done while not extrapolating the results of their study beyond the limits of their data. If the trial included evaluations of multiple outcomes, the potential for type 1 errors should be discussed. Conversely, if no significant associations with the intervention were observed, the authors should not interpret this as evidence of the truth of the null hypothesis. Factors involved in determining the external validity include the characteristics of the study units and study population, the trial setting, the interventions used, and the outcomes measured. Of particular relevance to veterinary research compared to human medical research is the applicability of challenge trials. Challenge studies use design features that reduce the potential for confounding and bias. However, these design features also have the potential to create important differences between the artificially induced disease study setting and the naturally occurring disease which occurs in real-world settings. As the authors make a general interpretation of the results, at a minimum, they should discuss results of the study in the context of all previous work, whether the results are supportive or not. Authors should avoid writing post hoc statements about the cost-benefit or cost-effectiveness of the intervention unless those economic endpoints were the purpose of the study and the methods for such analysis have been described as in items 6 and 12. To summarize, the reflex statement is intended to improve the reporting of randomized controlled trials in the livestock and food safety arenas. The statement can be applied to other arenas of animal research, including companion and laboratory animals. The increased utilization of the reflex statement should lead to complete study transparency by authors, which enables readers to understand a trial's design, conduct, analysis, and interpretation, and to assess the validity of its conclusions. As awareness of the reflex statement study reporting requirements increases, randomized controlled trial design, conduct, analysis, and interpretation should improve along with improved reporting. This presentation relied heavily on several papers, including the CONSORT Statement, 2001, the Revised CONSORT Statement, 2010, the Reflect Statement, and the Reflex Statement, Explanation and Elaboration.